Hello everyone, and welcome to our online lecture series for Bio2. Today, we take on our last lecture in our transmission genetics and Punnett square series. Because by this point, we've already discussed the structure of a genome, how it splits up in meiosis, the intricacies of reproduction involving meiosis and how meiosis can go wrong. We've talked about Punnett squares, their basics, dealing with dominant and recessive genes. And then we went much deeper, looking not only at a single set of genes, but pairs of genes and dihybrid crosses, and then talking about different inheritance patterns, whether traits are X-linked dominant, X-linked recessive, codominant, incompletely dominant, quantitative in their inheritance, or epistatic, amongst the others. We then discussed linkage between traits. And you'll recognize that we're revisiting linkage here today as well. Linkage was the idea that we only have some number of chromosomes in humans, 22 autosomes, 1 sex chromosome, but we have something like 30,000 genes. So each of our 23 chromosomes has to carry a large number of genes. This means that genes do not completely segregate or assort independently during meiosis. That independent assortment being the more important part here. Because ultimately you want genes to be shuffled around in meiosis. Remember, that was the whole point of reproduction, shuffling of genes. And therefore we looked at how recombination can play a role in shuffling genes when they do exist on the same chromosome. We measured recombination rates as percentage likelihood of recombining, or our centimorgan value. As a reminder, our centimorgan value looks at the number of recombinant genotypes. So in the case of looking at progeny, you're looking at how many progeny had a recombinant genotype versus the parental genotype. And this, of course, requires some test crossing. So hopefully those terms are familiar to you from last lecture. And we take the number of recombinant genotypes and divide them by the total number of offspring, multiply that by 100 in order to get a value that is representative of the percentage of recombinants that we'd expect, or the centimorgan distance. Now, centimorgans are named after the individual who came up with this metric, but ultimately it's just a way of looking at percentage recombination. So if A and B were 15 centimorgans apart on this chromosome right here, we would expect that 85% of the sperm made from an individual that was A1, or rather an individual that was heterozygous, having A0 and B0 on their other chromosome, all connected, that you would get recombination 15% of the time. So 85% of the progeny would be of the parental type, and 15% would be of the recombinant type. But we know that real life is not so straightforward. We know that when you have an expectation of 85% parental or 15% recombinant, that it doesn't always come out to exactly 85-15. For example, if you flip a coin, and we know that for the most part, or rather we assume that this coin is 50-50, 50% chance of getting heads and 50% chance of getting a tails. And if you flip it 10 times, are you going to get exactly 5 heads versus 5 tails? If you flip it 100 times, are you expecting 50 heads and 50 tails exactly? No, you might get something like 55 and 45, even if that coin is still actually 50-50. Today's entire lecture is going to be exploring that statistical variation in random draws in order to determine whether coins, or in this case, chromosomes, are truly 50-50, or if our reported distance of 15 centimorgans is actually accurate. In order to do this, we're going to utilize something called the Chi Squared statistic. Chi squared is written as the Greek letter chi, which kind of looks like an X, and then we square it. Now, the equation to calculate chi squared, we'll start out with the equation, and we'll reference it many times in this lecture because we will be doing some calculations, so please make sure that you have a calculator handy and are ready to pause the lecture as necessary. Our chi squared statistic is calculated by taking a sum. Didn't mean to erase the actual thing there. This is the Greek symbol sigma, which literally just means add up all of the things. And in our cases, our two things, we're always going to have two here, and they will be either the number of parental types or the number of recombinant types. And to be consistent for the remainder of this lecture, you'll notice that I will, hopefully, keep to the color assignments where numbers written in red are our parental types, written or highlighted, and numbers written or highlighted in blue are our recombinant haplotypes. And so we're going to be summing two different numbers, and our numbers derive from the equation of our observed number of progeny minus our expected number of progeny squared divided by our expected number of progeny in that class. So you will be doing this equation once for our parentals, in this equation once for our recombinants, and then adding them together to derive a value. So the explanation doesn't make much sense up until we try to walk you through a problem. So let's go ahead and start there. Our first problem is going to always need, or our first problem is going to be looking at loci that are unlinked. And in this case, our problem is going to be our null hypothesis. The null hypothesis is your idea here that things are as expected. Things work as they seem. And in our case, our null hypothesis are that our loci are unlinked. This means practically that they are found either greater than 50 centimorgans apart on the same chromosome or on different chromosomes. Because remember, if you are more than 50 centimorgans apart, you behave as though you're on separate chromosomes. And if you're on different chromosomes, well, we know that in meiosis you have a 50% chance of landing on the same side as the allele from the same parent. And you have a 50% chance of landing on the other side. In order to do this problem, we'll draw out our genotypes like this. You're always going to be needing to be given some information about parentage. So in this case, we know that our parents were true breeding, meaning that both parents were homozygous, not homozygous dominant, not homozygous recessive, but in this case, mom was homozygous A1A1, B1B1. Dad was homozygous A0, A0, B0, B0. Now, alternatively, be ready for questions where the parents could be true breeding A1, A1, B0, B0, and A0, A0, B1, B1. Or capital A, capital A, capital B versus lowercase a, lowercase b. These are all true breeding parents. You also can have this as a true breeding parent. Because what matters when you are true breeding is that you have homozygous genotypes at each separate locus. So I can change the letters, I can change the numbers, I can change the capitalization. Make sure you understand what it means to be true breeding. But back to our point, we know that our parents were true breeding, and therefore, when mom crossed dad in this case, they generated a hybrid baby. That means that the child got an A1 from mom, and a B1 from mom, and an A0 from dad, and a B0 from dad. And we are assuming that these are unlinked. If these are truly unlinked, we would expect that 1/4 of them would be A1, B1, as you see in this confirmation. And additionally, another 1/4 of them would be A0, B0, because if you are generating this side, then you're also at the same time generating that side, as we see here. Alternatively, if these chromosomes lined up so that the A0 was next to the B1 and the A1 was next to the B0, you would expect that 1/4 of the sperm or progeny would be A1, B0, and another 1/4 would be A0, B1. So with that understanding of the distribution of the different types of sperm that we're expecting to see, we can now go back and go forward rather, and make a table for our problem. We will be generating, I should recolor these, one sec, apologies. We will be generating different types or different counts of sperm here. So we'll be sequencing the sperm of this individual to identify how many types are parental and how many types are recombinant. So in this table, we see that A1B1 is a parental type. Whoops, did I already screw up the coloring? Of course I did. We see that A1B1 is a parental type, and we see that A0B0 is a parental type. And we see that A1B0 is a recombinant type, and A0B1 is a recombinant type. Now, looking back at our ratios, we're expecting 1/4 of each of them. So 1/4 of what? Well, that depends on how many sperm we genotype. So we have to have a total here. Our total is going to be derived from our observed number. Let's say hypothetically that for this question, we sequenced 500 sperm in total. And of those 500 sperm, 126 of them were A1B1. 124 of them were A1B0. 126 of them were A0B0. And 124 of them were A0B1. In order to calculate our expected from our observed 500 sperm total, What we need to do is take the percentage that we expect to see in each of our categories and multiply it by the total. This means that our expected here is 500 times 1/4. Well, 500 times 1/4 is equal to 125. So we would have expected 125 sperm to come up A1B1 if these loci are truly unlinked and found on separate chromosomes. We also would have found 125 of our A1B0, 125 of our A0B0, and 125 of our A0B1. In the case where we're looking at unlinked loci, you can see that our expected values will be the same. But remember, every single time that we make one recombinant, we make two. So when we are looking at our numbers, we do not want to separate them into 4 categories. We instead want to separate them into the 2 categories of parental or recombinant. This means that we have to identify our parentals and add them together, and identify our recombinants and add them together. In this example, we see that we have 252 parental types and 248 recombinant types. So let's look back at our equation. We needed some observed numbers for parental and recombinant, we needed some expected numbers for parental and recombinant, and then we need to plug it into this thing. So let's bring this thing down with us so that way we can keep a visual on it. And let's start writing. So our first term of our sigma is going to be for our parental types. Our observed number was 252. We're going to subtract that from our expected. Now keep in mind, our expected is also a term that gets added together. We had 125 of these and 125 of these. Therefore, our expected is actually 250. We're going to square that value and divide it by our expected, which was 250. So in this case, you're looking at 252 minus 250, or 2. 2 squared is 4. 4 divided by 250 is 0.16. Please make sure that you can enter this correctly in your calculator. I don't care what you prefer to use, but I do care that you are able to enter things with syntax correctly in order to make sure that you are calculating things correctly. So our first value is 0.16 for our parentals. Our chi-squared value for our recombinants is 248 minus the 250 expected for our recombinants, because again, we expected 125 of these, 125 of these. Bam, that makes 250. We're going to square that value, divide it by 250, and we find, hopefully again, able to enter it into the calculator, you find that that value is 0.16. Now keep in mind that our chi-squared needs to be a sum of these terms. So, before you move on, and this is a mistake I commonly see, make sure that you add the 2 terms together. You got a 0.16 from parentals, a 0.16 from recombinants. This gives you your final chi-squared value of 0.32. Okay, so what's that mean? Right? So we've arrived at our final chi-squared value. That's a number that means nothing to me unless I start to understand what the statistic entails. What's underneath the hood. What does 0.32 mean? In order to understand what 0.32 means, we have to look at the chi-squared distribution. Meaning that we're looking at a curve. Now, on this curve, you'll notice that the y-axis is a probability value, and the x-axis is your chi-squared value. So, what this means for me is that there is some likelihood that my value that I calculated, which was 0.32, which 0.32 is going to be somewhere around here. It's a very low value. There is some likelihood that even if my values were drawn from a 50-50 distribution, or a 50-50 coin flip, or unlinked loci, that they might vary away from that. You notice that the values here, 126, was not really different from 125. I cooked the books on these. I've just selected all the numbers today on purpose to illustrate different points. You notice that there's not a lot of variation between the 252 and the 250 observed versus expected parental types that we had. And therefore, you can kind of say with fair certainty that, like, yeah, you know what? Those are probably unlinked. 126 is not that different from 125, and 126 is not that different from 125. Looking at them together, 252 isn't that different from 250, so therefore it's probably pretty likely that those are unlinked. But we want a mathematical measure of how likely that is, because there will be some cases where the area is gray, where you don't see so readily as to whether loci are linked or not. Chi-squared allows us to do that by looking at where this value lands on the distribution. The way scientists do this is they say that we will accept any chi-squared value that describes 95% of the area underneath the curve. That means that you had a 95% chance of generating your chi-squared value from a 50-50 distribution as long as it is somewhere below a threshold. And you can see that this threshold of 5% tail, meaning that it is in the tail end or a lower likelihood of being pulled from that distribution if it's found out over here. So again, explaining it another way. 95% of the time, if you randomly pull sperm counts from truly unlinked loci, you're expecting to get some chi-squared value in here. But 5% of the time, you'll get a chi-squared value that lands outside of that major density underneath the curve. And if you land in this tail, that means that you are probably not drawn randomly from that distribution, and therefore we should reject the null. And in this case, that null being that those loci are unlinked. If you got sperm counts that were so skewed that you did not get a value landing in this 95% area under the curve, that means that it's probably likely that your loci are actually linked. The value that is our cutoff for 1 degree of freedom, and I won't be going into what 1 degree of freedom means, but it's a statistical concept that I encourage you to go into further if you take a stats class, and you definitely should take a stats class. Our chi-squared value for 1 degree of freedom, our threshold, this spot, is a chi-squared value of 3.84. That is always the value that we're going to be using. If you have a value less than this, that means that you accept the null. And if you have a value greater than this, that means that you're going to reject the null. And you can see a full table of chi-squared distribution or chi-squared values from the distribution based on different degrees of freedom and based on different thresholds. So scientists can have different p-values, and our p-values we'll refer to as alpha. For purposes of this class, our p-value or alpha threshold is 0.05. That means 5% of the time the value lands far out. If you want to be a little bit more conservative, you could go with a p-value or alpha of 1% of the time or 0.1% of the time. But you will see by reading this table that if you have 1 degree of freedom and we're looking at a p-value of 0.05, that our chi-squared threshold is at 3.84. So, with a value of 0.32, our value is less than 3.84. Therefore, in this case, we conclude that our loci were unlinked given these observed sperm genotype ratios. For the rest of the lecture, we're going to keep going over different problems and looking at things in different ways, because you will be expected to solve these problems on your own. So next up, let's fill in some values for another chi-squared problem, looking at the exact same question. Are these loci linked or not? With our null expectation being that they are linked. Are not linked. So, and the reason why you have to go with not linked is because that gives you that null expectation of an equal distribution of your parental and recombinant haplotypes, where you're looking at a centimorgan distance of 50 centimorgans. So, in this situation, we record, again, from same style of cross, looking at parents that were true breeding for the A1, A1, B1, B1, and A0, A0, B0, B0. So just go ahead and copy down that this is our parents again. And here we're looking at the sperm of our progeny. So you're going to be observing these haploid genotypes. So our observations state that we found 150 A1B1s. We found 110 A1B0s. We found 130 A0B0. And we found 110 A0B1s. And again, looking at these in terms of parental, we see that we have a total of 280 parental types, and we see that we have a total of A1B0 recombinant, A0B1 recombinant, 110 and 110, or 220 recombinants. Now, what are expectations if these are unlinked loci? Well, that means that you would expect to have the exact same number of A1 B1s as A1 B0s, as A0 B0s, as A0 B1s. So let's take a look at our total here. Well, 150 plus 210, that gives you 260, plus 130, that gives you 390, plus 110, gives you a total of 500 of these. So that means that you want to split 500 into 4 categories, or 500 divided by 4 is 125 of each of these. But remember, for our chi-squared, we're using that 1 degree of freedom, meaning that we need 2 different categories, so we're grouping things based on whether they are parental or recombinant. That means that our expected is actually to have 250 parental, 250 recombinant, and let's see if these values agree with that distribution. So switching the pen color here to red, let's do our chi-squared equation again, and just for ease here, I'll scroll on up and we'll copy this down, or rather copy the equation, because you'll always have access to the equation. You'll have your notes with you. So always keep this handy. You can see that it's observed minus expected squared divided by expected. So observed for our parental was 280 minus expected of 250. Square that total, divide it by the expected of 250. Add this value to our observations for our recombinants. Well, our observed recombinants were 220 minus the 250 expected, square that, divided by the 250 expected. Well, this plugged into a calculator, 280 minus 250 is 30, 30 squared divided by 250 gives you a total of 3.6. Same thing over here, 220 minus 250 is 30, 30 squared divided by 250 is 3.6. Now, this is where some of the more common mistakes occur. A lot of folks tend to go, oh, 3.6, 3.6. Is that less than my threshold of cutoff of 3.84? Well, 3.6 is less than 3.84, and therefore we're good. This is not a situation where these loci are linked. They look just like we would expect. They fall within the 95% confidence interval, and we're good. But that's not true here, because you have to remember to add these two values together. 3.6 plus 3.6 is 7.2, and 7.2 is greater than 3.84. Therefore, our conclusion is to reject the null and assume that these loci are linked. Does that make sense? 7.2, the value calculated from calculating our chi-squared distribution, ended up being greater than our cutoff. Therefore, we see that we should reject this and assume that these loci are linked. All right. For our next problem, we're going to switch things up a little bit. Rather than assuming that the loci are unlinked, I'm going to give you the fact that the loci are linked and that their distance is 20 centimorgans apart. So we assume that they are linked together and that they are 20 centimorgans apart. So I want you to determine, based on our observations, whether I am lying to you about them being 20 centimorgans apart. So what will this do to the structure of our problem? Well, previously, when we calculated our expected totals would be the distance they are apart times the number of progeny. And this value would change because you're either looking to bin them into 4 different groups or have some sort of distribution, right? So in the case where everything was 50-50, you would try to group those two distances into 0.5, 0.5, and then again divide them into the parental and recombinant of each type, where we see, one parental being here, the other parental being here. This is why you would get 125 and 125 in our previous problem. And then with the recombinants, you'd expect the same thing, right? That they're also grouping into these 2 different categories, and therefore you should expect equal proportions here. But my naive expectation with this problem is that you are not given equal proportions. There is going to be a bias in terms of your parental and your recombinants. You see the parental and the 2 different parental haplotypes, with A1 being linked to B1 and A0 being linked to B0. So you know that, again, we keep our consistent situation here with the 1 subscripts connected together being parental and 0 subscripts connected together being parental. And then when we mix and match 1s and 0s, those are our recombinants. So that gives us the ability to figure out how many we are expecting of each class, assuming they are 20 centimorgans apart. So now let's figure out what are those expected numbers once we get our observed numbers, because that gives us the total number of progeny. So we have 288 observed of A1, B1, 60 observed of A1, B0, 275 observed of A0, B0, and 57 observed of A0, B1. So first thing we want to do here, we want to get the total number of progeny. So we add 288 plus 60 plus 275 plus 57. Again, you'll have a calculator. This adds up to being 680 progeny. So now, move this, scoot this just a bit over here so that way we have our space. How do we figure out what the expecteds are for this? If you have 680 progeny and you expect the parentals and recombinants to be 20 centimorgans apart, what that means is that 680 is going to be divided into 80% parental and 20% recombinant. Now keep in mind, you are given these 4 values, but we can again identify the 2 recombinants. 57 plus 60 gives us a grand total of 117 recombinants. And on our parental side, 288 plus 275 gives us 563 of our parental type. In terms of our expected values for parental and expected values for recombinant, well, all you got to do is multiply the 80%, or 0.8 times 680 to get your expected parental quantity, which gives you 544, and then 0.2 or 20% times 680 to get your recombinant types, that are the total number of recombinant progeny that are expected, or 136. You can always double check your work by making sure that 544 plus 136, yep, that adds up back up to 680. So now we have all of our values. We have an expected amount of parental types of 544 altogether, and an expected amount of recombinant types of 136. So let's go ahead and put together our chi-squared equations again. So observed minus expected, we'll start with our parentals. Observed was 563 The 3 needs to look more like a 3. Minus the expected number was 544. All of that squared divided by 544 expected. Add that together with our observed number of recombinants, 117, minus expected, 136. Square that whole thing, divide it by our expected of 136. So, solving these out separately, entering them into a calculator. Again, give yourself a moment here. Please make sure that you know how to enter this successfully either into the Google search bar, because that's also a calculator, or into your own handheld calculator. You'll find that the chi-squared value for just the parental types is 0.66, and then the chi-squared value for our recombinant types is 2.65. 0.66 plus 2.65 gives us a value of 3.31, which ends up being less than 3.84, our chi-squared threshold of 0.05. And because it is less than our threshold cutoff, going back to our graph here, even though that value is somewhere close to the cutoff, it still falls between the cutoff and 0, meaning that it is within this 95% expected value, meaning that we cannot reject the null. We accept the null, meaning that these loci are actually 20 centimorgans apart, or at the very least, that these observed number of progeny would be consistent with two loci that are 20 centimorgans apart. Now note that if you go back and try to calculate the exact distance of centimorgans between these progeny, you may or may not get exactly 20 centimorgans apart. So as practice, let's do that real quick. Change color here so that way it really sticks out. But if I were given these observed values for progeny, how would I calculate the distance between A and B as loci? Well, looking back at your notes, hopefully you remember that the number of recombinants divided by the total number of progeny can give you that centimorgan distance. And in this case, we know that our total number of recombinants was 117, and our total number of progeny was 680. 117 divided by 680 gives you 0.172, or a centimorgan distance of 17.2 centimorgans. So you see that while I told you for sure that these loci are 20 centimorgans apart and the observed number of progeny falls in line with that observation, you cannot reject the null there, that if you use this to actually calculate a direct distance from these progeny, that you would instead get a slightly different value. So these are just some of the many things that you hopefully are able to do after these series of Punnett square and linkage lectures. So let's end by doing a broad review of the different types of problems you should be able to solve. So first and foremost, you have to be able to solve mode of inheritance Punnett squares. Oops. What this means, let's take a moment to talk about each of these really quick, just verbally go over a problem. I do hope that you actually can generate some of these problems yourselves, so that way, because the best way to understand a problem is to try to write one, and then exchange with classmates. That's historically been the best way to study for these. So what I mean by solving mode of inheritance Punnett squares is if I give you some genotypes, you have to tell me, genotypes and phenotypes, you have to tell me whether The genes involved here are codominant, incompletely dominant. Is there quantitative inheritance? Is it simple dominance and recessivity? What is the mode of inheritance given different genotypes? Number 2, you have to be able to calculate expected progeny ratios. What that means is that you have to be able to calculate how many progeny of either parental or recombinant types you expect to see with given distances. So that is exactly what we did as part of our problem just above. Given a centimorgan distance, you can calculate expected progeny ratios. Now, you can even simplify this even further by actually looking at some of those simple mode of inheritance Punnett squares and seeing how many progeny you expect to be of these different genotypes. So, for example, that might mean taking like a dihybrid of two different genes, like pea plants that are tall and short versus, or plants that are tall and short and have peas that are green and yellow, and giving the expected ratios you'd expect to see given two true breeding parents or something in the F1. So stuff that you've done either in association with linkage or not. Number 3 that I'd certainly expect for you to be proficient in is calculate centimorgan distance between linked loci using progeny ratios. Oh, that W is pretty bad, huh? And that's what we did just a moment ago when we looked at, okay, how many different progeny we have, how many of them are of the recombinant, how many of them are of the parental, and how many of them are total to calculate a centimorgan distance. This is looking at a direct measure rather than looking at whether it is statistically different from a direct measure. Number 4 is determine if the loci are linked using progeny counts. And so that means going back to our first problems of where the null expectation is that they are unlinked, and then you are using observed counts and expected counts, where the expected counts are all 50-50, and seeing whether you can reject or accept the null hypothesis there. So that's something that we've done a fair bit of. You'll notice that the determine if statement is where you're going to be using chi-squared values, because our next and final type of problem that I'd expect you to be able to solve here is determine if the centimorgan distance is accurate. Between linked loci, given progeny counts. And so again, with that, if you are, instead of using a centimorgan distance of 50-50, you're given some sort of biased value, 80-20, with 20 centimorgans apart, and calculating the chi-squared value for that. So broadly speaking, in terms of the challenge areas, you will have one challenge area that looks at calculating progeny ratios with modes of inheritance, so where you don't know how things are inherited, but given numbers of progeny with different phenotypes, you can calculate those. And that's where I'll complicate things with quantitative inheritance versus codominant inheritance versus epistatic interactions between the alleles and the loci, or multiple alleles in the first place. So you have all of those different modes of inheritance, and that's one of these difficult pillars. And then the second pillar is looking at chi-squared values, whether that be with centimorgan distance or unlinked loci, and that kind of problem. So either which way, Punnett squares are fundamental to all of these questions, and a basic understanding of how to determine haplotypes. Is going to be critical. You can't solve any of this without understanding haplotypes and how loci are linked. You can expect to find some practice problems listed on the course webpage, so that way you can get some practice here. But again, I would encourage you to look back and try to design your own problems and exchange them with classmates. Practice does make perfect on this, so please give it a try.